very much to be with us today. In the name of all the students and professors from our graduate program in biophysics and from the Carlos Chagas Filho Institute of Biophysics from the Federal University of Rio de Janeiro, it's a pleasure to have you with us today, and we have the pleasure to sharing with us and hearing about your excellent work. Thank you very much. And please, Professor van der Leij, could you introduce our professor?
Yes. Well, it's for me a great pleasure to introduce Professor Wolfgang Baumeister. He made got his undergraduation in biology and biophysics at Münster University and Bonn University. Then his PhD in Düsseldorf with a postdoc at Cambridge University. And then in the Max Planck Institute in Martinsried, he developed an excellent work in cryo microscopy, apply it to develop many techniques and apply it to different biological systems. He's member of several academies, including the German Leopoldina Academy and the United States National Academy of Sciences. Was editor-in-chief of the Journal of Structural Biology and Biophysics and Biochemical Research Communication, and member of the editorial board of several journals in the areas of protein, microscopy, cell biology, etc. Is distinguished professor at the Caltech Institute in California and received several prizes. I will mention only a few, like the Ernest Ruska Prize, the Otto Warburg Prize, and the Harvey Prize from Israel. He has published a large number of papers, around and most of these papers published in journals such as Cell, Science, Nature, PNAS, and so on. With, according to the Google Scholar, you have 52,000 citations with an H index of 120. Indeed, Professor Baumeister is really one of the most important microscopists today. His work is well recognized. And what is important, he's always participated of the congresses in Latin America. He came to Brazil 1 or 2 times, in Cuba and in other places of both the Brazilian Society of Microscopy or the CIASEM meetings. And so that he likes to not only to work in his lab, but also to enter in contact with colleagues from different parts of the world. Today he's going to talk us about structural biology in situ, or the power of seeing the whole picture. I enjoy very much the title. That's it's very innovative title. So, Wolf, really, it's a pleasure to have you here in this web symposium. That the people say first web symposium. I think that it will be only this one. I prefer the classical symposium, so that you can start your lecture.
Okay, thank you very much, van der Leij, for your kind introduction. Thank you for inviting me to give this lecture on occasion of the 2 anniversaries of the 2 institutions in Brazil. It would have been much more fun if I could be in Rio and give this lecture in person, but well, the current situation simply does not allow that. So, as Vandali said, the title of my talk today is Structural Biology in situ, or the power of seeing the whole picture. Now, let me begin by explaining why we want to do structural biology in situ at all. I think traditionally, structural biologists take a reductionist approach. Cells are taken apart, the molecular components are fractionated, they are isolated, purified, and then they are studied one by any of the established methods of structural biology, X-ray crystallography, NMR, and increasingly EM single particle analysis. Now, this divide and conquer approach has been very successful if you look at the PDB, but it's also true that very rarely biological functions, cellular functions can be attributed to individual molecules. Cellular functions arise from the interactions between the many molecules that are found in the cell, which are acting somehow in concert. So structural biology in situ aims at studying macromolecular, supramolecular structures in their functional environment that is inside unperturbed cellular environments. Now, the method enabling to do structural biology in situ is cryo-electron tomography. It's the application of tomographic principles of data acquisition and reconstruction to samples which are embedded in a frozen hydrated state. So the only preparation step is essentially rapid freezing. So I think you are going to yield a 3D image of the structure under study. So cryo-electron tomography combines the power of high-resolution three-dimensional imaging with the best possible structural preservation of cellular structures. So what you see, well, that is the principle of tomography in terms of data acquisition, in terms of reconstruction. And on the right-hand side, you see a first example of a tomogram basically showing a mitochondrion in close vicinity of the lipid droplet in the HeLa cell. Now, the idea to do electron tomography is not new. A quite remarkable paper was published back in 1968 by Roger Hart. He was at Lawrence Livermore Laboratory. It was entitled, it was published in Science the same year, actually almost the same time when Aaron Klug and David Rosier published their famous first 3D reconstruction of a phage tail. It's entitled Electron Microscopy of Unstained Biological Material, the Polytropic Montage. So the polytropic montage is synonymous to tomography. So the abstract says with the use of an electronic picture scanning device, which didn't really exist at the time, and a digital computer, electrons micrographs taken of a specimen along several different directions can be superimposed to form a montage that is more informative than the component images. Preliminary results indicate that one may thus study unstained, unshadowed biological material at high resolution. So the vision was there, but the technology was not there. I think Roger Hart was on the right track, but I mean, he passed away at a relatively young age, 3 or 4 years after he published that paper. So now, I think the main problem and the reason why it has taken another 20 years basically to come into existence is basically the fact that biological material embedded in ice is extremely radiation sensitive. And tomography, of course, involves multiple exposures. Typically, let's say 100 or so images have to go into a reconstruction. So we began about, in the late 80s, as a matter of fact, to develop automated procedures for data acquisition, allowing to keep the electron dose within tolerable limits. So basically, I think in the second paper at the bottom, we could say that it is demonstrated that the cumulative electron dose in collecting tomographic 3D datasets can be kept small enough for the investigation of ice-embedded specimens. Now, historically, this was the first tomogram ever taken of a biological sample embedded in ice, something as trivial as lipid vesicles in a thin layer of ice. That was 25 years ago. Now, for quite some time, there was another limitation, and was the size of the thickness of the sample. So tomography was limited basically to relatively small objects, such as viruses or small bacterial cells. But even bacteria, I mean, which are typically something like 1 micron or so in thickness, are at the limit of what is, that is, well, almost 20 years ago. Now, I'm showing it for a different reason, and that is because of the dose limitation. Tomograms are still fairly noisy. You see in the top row basically some examples of the surface of this virus particle, and you see the images are noisy. Now, whenever a tomogram contains repetitive features, one can apply some averaging. One can computationally extract these repetitive structures, one can align them and can average them, and then one can, the improved averages, I mean, put back precisely into the context. That is what is done here in this case with, I mean, the nucleocapsid here, that is the only repetitive feature here. These glycoproteins here on the surface, they are highly flexible and cannot be averaged. But I mean, here we have the nucleocapsid, and we can replace the noisy density by the improved average density. Now, as I said, I mean, a limitation for quite some time was to deal with specimen thickness. Cellular structures, eukaryotic cells are all by far too thick to be studied in toto. Only, I mean, the periphery or appendages were accessible to tomography. Now, the possibility is, of course, cryosectioning, as was tried by Jacques Duboucher and others. But the problem is that vitreous ice is highly compressible. And I mean, you induce very severe artifacts if you try to physically section vitreous ice. I mean, I'm showing here an example. We have an interest in the structure and the molecular architecture of the so-called rod outer segment here of rod photoreceptor cells. Now, I mean, they are about of the dimensions that can be dealt with, and we had a first go at that something like 15 years ago. Now you see the isolated rod here on the right-hand side, but you see already that only, I mean, the left-hand side, which is a bit flattened, is sufficiently thin to be transparent to the electron beam. So we invested quite a bit into an alternative method of thinning. So what you see here are cells, dictyostelium cells, grown directly on an EM grid. And we used focused ion beam milling for thinning. So what we do essentially is we try to cut compression-free windows into cells. So we use a dual beam instrument as shown here. So we have an SEM beam, which allows us to monitor what we are doing. It gives us an idea where the object is sitting. And then we have a focused ion beam, mostly these days, but this is changing rapidly. It's a gallium beam that is used then to ablate material. Now, different geometries can be used. So again, you see at the top, basically a cell sitting on an EM grid, and we can ablate material from above and below. And what we cut is essentially a thin lamella, as we call it, which is supported on the left-hand side and on the right-hand side only by anything between 100 nanometers and 500 nanometers, depending on, I mean, the structure that has to be accommodated. Now, I should say now, before, I mean, showing applications, in this case, to the rod outer segment, I mean that in recent years, we all know there have been enormous advances in EM technology. I think the latest generation, I mean, it's not so recent anymore, of microscopes has a lot of robotics, increasing throughput massively. and also ensuring consistent quality of the data. A game changer were the direct detectors, much superior in terms of detective quantum efficiency to CCD cameras. But there are also small devices like the faceplate, which allow to generate phase contrast in focus. I mean, to very briefly, I mean, if you take an image of biological material embedded in ice in focus, you get an image like the left-hand image, which has basically no contrast. So contrast is generated by defocusing, but that results in a pretty weird contrast transfer function, resulting in these kind of ghostly images, in this case of Fritz Zernike, who invented the faceplate for light microscopy. So the dream was always to have a faceplate for electron microscopy, and I think one solution, not the ultimate solution, but a solution is the so-called Volta faceplate developed by Radoslav Danev in the lab. It enables us to take images in focus with sufficient strong phase contrast. It's basically a thin carbon film. The phase shift is generated by a beam-induced Volta potential. So it's a fairly practical solution to this problem. Now, if we go back now to the rod outer segment, now this is of course after FIP milling. So I mean the sample is something like 300-400 nanometers now in thickness. On the left-hand side, it's a typical defocus image generating phase contrast. And you see the much improved contrast obtained with the faceplate in focus. Now that is important. I think you can squeeze out a lot of information from the left-hand side image if you can do averaging. If you can't do, I mean, then I think there are many features which remain hidden in the noise. So like in this particular case, I mean, you will see here, we were interested what maintains the architecture, the supramolecular architecture of the membrane system with the precise stacking of these membranes here. And now we see very clearly there are certain spacers. There are two types of spacers. One on the left-hand side near the periphery. There are other spacers, I mean, further inside between the disc membranes. Now, that is just to illustrate basically how these two methods, focused ion beam milling and I mean in focus phase contrast, allow us to see and to detect features which were otherwise invisible. Now, I will run through a few slides here. That's work of Julia Mahamid. Top left, I mean, you see an HeLa cell sitting on an EM grid. What is stained here is the nucleus. Now we can target that very precisely. Bottom right, you see a thin lamella cut at the periphery of the nucleus here because we were interested in the structural organization in the nuclear periphery here. So now we can take a tomogram of such a lamella walking from inside the nucleus at the top. So we start somewhere in the heterochromatin, and what we see on the right-hand side here, I mean, are individual nucleosomes, so we can depict them clearly. We can extract them and we can average them and get a resolution of, let's say, something like 12 angstroms in this case. But I mean, what is more challenging and much more interesting than, I mean, the well-known structure of the nucleosome is, I mean, their connectivity. And that is still a major challenge in segmentation of such images, of such tomograms. Now, as we walk from the inside to the outside, we approach then next the nuclear envelope, the inner side of the nuclear envelope, which is covered by the meshwork of the nuclear lamina, a thin intermediate filament, fairly disordered meshwork, but very important for structural maintenance, but also for interaction with the chromatin. Now we have selected a slice where we see the nuclear pore complex, first the nuclear side, and then, I mean, as we walk outside, I mean, the cytoplasmic ring of the nuclear pore complex. In the bottom line, I mean, we have arrived now on the outer surface of the nuclear envelope that is studied with ribosomes, and you see very easily, you see actin filaments, you see microtubules, you see that the ribosomes form relatively well-defined polysomes here on the surface. Now we can do, of course, some image processing, segmentation of microtubules, actin, the nuclear lamina. Now at the bottom what you see is some averaging of the nuclear pore complex. I think the structure you see here of the nuclear pore complex is not yet a very high resolution structure, something like 30 angstroms resolution, but it is remarkable in the sense that it has been obtained from only 5 copies of the nuclear pore complex in this particular case. So, and that is due to the fact that, I mean, there is this very obvious improvement in contrast due to the phase plate. Now, why is that important? It's important because alone of the nuclear pore complex can change by something like 13–15% or so. So I think ideally we would like to push it far enough that we can get a structure of that resolution, I mean, from a single nuclear pore complex. Now, I think this is now the synopsis of that particular experiment. I mean, you see again in the background the chromatin, then you see in this reddish color, I mean, the meshwork of the nuclear lamina, and on the surface you see the polysomes, etc., etc. Now, the next couple of slides come from a cell which we like a lot. It's because it's an algae, Chlamydomonas reinhardii. The advantage of this cell, let's say, compared to a yeast cell, is that both the cytoplasm and the nucleoplasm are a little bit less crowded. That facilitates the image analysis. The second advantage of Chlamydomonas is that the organelles in this cell, I mean, have a well-defined topology. So it's very predictable where you find a Golgi complex, etc., etc. That facilitates this. It's illustrated on the right-hand side. So you see basically here the nucleus, and you know that, I mean, there are 2 Golgi complexes in the cell, and you see on the left-hand side in this movie, basically it focuses here on one Golgi apparatus. Here is some endoplasmic reticulum studied with ribosomes. Here is the nuclear envelope again, and this is the position roughly of a nuclear pore complex here. Now, in this case, it was a bit easier and we could do quite a bit of averaging. I will show only a few examples. There are dozens of structures here from COP1 vesicles, etc., where we have obtained very nice structures. I mean, the next one shows, I mean, oops, this was a little bit too fast. The next one shows the nuclear pore complex, now with a better resolution. I mean, in red, that is the so-called Y complex, which is a highly conserved element, but between species, the oligomeric state can vary quite a bit. So this is, in this case, we had something like 100 or so nuclear pore complexes, so resolution is already quite a bit better. The easiest to deal with is always ribosomes. Ribosomes are abundant because, I mean, one limitation, this is the difference from tomography to single particle analysis. In single particle analysis, basically, you have as many copies of a molecule as you like. In the case of tomography, the number of copies, I mean, is dictated by nature, by the natural abundance of the molecule. But ribosomes are always abundant, and so without pushing the limits, I mean, a resolution well below 10 angstrom can be achieved. The next one shows now a ribosome sitting on the endoplasmic reticulum, sitting on the translocon, the protein conducting channel SEC61. And you see here, I mean, as you zoom in on the translocon here, that resolution is now in the range of something like 7 angstrom. So the helices of the translocon are very clearly visible. Now, I think I have to explain a little bit the technique we use to make lamella is quite suitable if you have cells which are grown multicellular organism, or if you want to deal with, let's say, there's some tissue. So I think I go back to this movie here. So in this case, we wanted to target a cell in the gut of C. elegans, and it's guided by fluorescence microscopy, so we know where it is. But we have to cut deeply, vertically now into the worm in order to target these cells. And then we have to cut that free, that block of ice, which may be something 3-4 micrometers in thickness. We have to transfer it to another holder to ablate more material, and then we can take a tomogram. You see the quality is roughly the same here now with this lift-out procedure, which is becoming also commercially available now. You see, I mean, so we have the gut here. We have, I mean, on the right-hand side here, we have, I mean, coated vesicles. Here we have a piece of ER studied with ribosomes. This is a cross-section of a microtubule. So resolution is fairly good. Again, we can do now subtomogram averaging. We can, given the quality of the data, I mean, we can distinguish between ribosomes at different stages of the elongation cycle. We can judge that from the occupancy with tRNA. And I mean, so the two different stages we can map back to the tomogram, as done here and shown by the yellow and blue colored ribosomes here. Now, I should say that we have only a limited interest in ribosomes. Our interest is on the protein degradation side since many years. So the main player in the cell there is the proteasome, which is a 2.5 megadalton complex built of 34 different subunits here. Now, on the right-hand side, you see a structure which here produced something like 7 years ago. At that time, resolution was, that was of course ex situ to start with. I want to use the ex situ data to introduce the system. Resolution was something like 7 angstroms, so not sufficient for a molecular interpretation, given also the fact that, I mean, we have 6 homologous HPA subunits, etc., etc. So at the time, in order to interpret it in terms of molecular architecture, we had to use an integrative approach guided by these strains, which allowed us, based on the 7 angstrom model, I mean, to come up to determine the molecular architecture of the 26S proteasome. I think many of you will know that it operates at the executive end of the so-called ubiquitin pathway. So it degrades proteins marked for destruction by ubiquitination. Now, I think I will walk you quickly through the basic architecture. It comprises a core particle where degradation takes place in red, and that contains, I mean, a cavity inside. So any substrate to be degraded has to be translocated into this cavity where the active sites are located. And there is a gate controlling access to that. Now, in order to become functional, the 20S core particle has to team up with a regulatory particle. Their role is to prepare substrates for degradation. That means recognizing ubiquitinated proteins, deubiquitinating them, then unfolding them, because only unfolded proteins can be thread through the gate that controls access to the core particle. So I think we have now pretty high resolution 3 angstrom structures of the 26S proteasomes in different conformational stages. I mean, it's a highly dynamic structure, and you will get a glimpse of the dynamics in the next minute or so. So that is the so-called ground state, in which, I mean, it is not dealing with substrate, the prime state. It has bound substrate, and the process has become irreversible. But then, I mean, there is the substrate processing state, which means, I mean, now the substrate is unfolded by the AAA ATPase module, which is shown here in bluish color. and also the ATPase uses the ATP, the energy derived from ATP hydrolysis, to push it into the core complex for degradation. So there are many conformational changes during the functional cycle. They serve to bring the deubiquitylase in position for deubiquitylation. They align the unfolding channel in the ATPase with the gate of the core particle. So we have fairly large-scale conformational changes. I mean, rotations of whole ensembles of subunits by something like 25%, shifts of something like 15 angstroms or so for alignment. Now, returning to the in situ situation. So what you see here on the left-hand side are light micrographs of in green are proteasomes are in the nucleus. And then there are two locations here, I mean this puncture here, where we have a fairly massive accumulation of proteasomes, and that is in the vicinity of the endoplasmic reticulum. So we can again zoom in and take our tomograms. So first we look at the proteasomes inside the nucleus. So you see here on the left-hand side, I mean, you see the double membrane of the nuclear envelope, the position here of the nuclear pore complex, and you see even with your naked eye, I mean, basically the structure of the 26S proteasome, the core particle and the regulatory particle. Of course, they occur in all different orientations, and so we have to search for them systematically, but we can do that with a very primitive template, essentially just a cylinder of the right dimensions, and we can do the subtomogram averaging, yielding us now the structure basically, of course, at lower resolution in this case, as the ex situ structure here. But we can do quite a bit of classification of proteasomal states. We can distinguish between proteasomes which have one or two regulatory particles, so they're assembly states. We can look for the basic conformation, and we can distinguish between the ground state shown here in green and the substrate processing stage shown in magenta. And from that, we can infer whether a proteasome is degrading substrate or is sitting there and waiting for substrate. We can also determine whether they are free or whether they are bound to other cellular structures here. I don't want to go into that in great detail or in terms of numbers, but the nice thing is that we retain the information where each of these proteasomes comes from. So this is, again, a tomogram here of the region. We have basically at the center, we have the nuclear envelope. On the right-hand side, we have the mitochondrion sitting in the cytoplasm. All proteasomes are first in light blue. Then we can distinguish the ground state in green and those that are substrate processing in the bluish form. Now, we can take a closer look at that. I mean, you will see that many of the proteasomes are sitting in close vicinity to the nuclear pore complex. And when we do a deeper analysis here, basically we have two subpopulations. We have one group that is sitting relatively close to the membrane level, but at the periphery of the nuclear pore complex. We have a second population that is further towards the center of the nuclear pore complex, but much further away from the level of the membrane. And that is the position of the so-called nuclear basket. So we can see that with individual nuclear pore complexes here. I mean, the yellow ones contacting the nuclear basket, and the reddish one, contacting the membrane at the periphery of the nuclear pore complex. So I think we are identifying currently the tethers that link them to the nuclear basket. We assume we have some evidence that those, I mean, are involved and inner membrane of the nuclear envelope are continuous, but they maintain a distinct protein composition. And that is done basically by degrading or removing proteasomes from one side. Now, there's a second hotspot, as I indicated before. The second hotspot is, I mean, associated with the endoplasmic reticulum. It's a well-known, it's a huge field in protein quality control in the cell, ER-associated degradation, or ERAD. Basically, it is a pathway targeting misfolded proteins in the ER for ubiquitination and subsequent degradation. So basically, the scenario is the following. Ribosome sitting on the endoplasmic reticulum synthesize protein. The protein is translocated into the ER, where it's supposed to fold properly with the assistance of chaperones. But that can go wrong, especially under stress conditions. And then we have protein aggregation, and that triggers the so-called unfolded protein response. There's no powerful degradation system inside the endoplasmic reticulum, so what is needed is basically a retrotranslocation of the unfolded, misfolded proteins, and then, I mean, it's taken over by the proteasome plus amino AAA ATPase CDC 48 for degradation outside the endoplasmic reticulum. That is the basic pathway. Very well studied, hundreds of publications. But let's say the molecular organization was not visible so far. As we zoom in on endoplasmic reticulum, shown here in gray, we see, of course, I mean, thousands of ribosomes sitting there. The dark blue ones are those directly sitting on translocons. The light blue ones are free ribosomes. And the clusters in red, comprising typically something like 30 or so particles, these are the 26S proteasomes sitting, forming clusters on the surface of the ER. Interestingly, always on the side opposite to the site where, I mean, vesicles are secreted from the ER, so that it's well separated. Now, let's zoom into that. I think we have to do some sort of molecule by molecule analysis of the tomograms. We can, if we have, I mean, high resolution structures, we can use them as templates. But what we have done here is basically we picked all the particles in the vicinity of these clusters here, and then run through some classification, sorting them according to similarity into separate classes, and did subtomogram averaging here of these classes here. Now you see the result here basically. So we use, I mean, a template-free approach. It's really easy for the proteasome, and we got, I mean, a decent structure here. Ribosome, very easy to get a decent structure. CDC48, I mean, is tenfold less abundant even in these clusters than the other molecules, so resolution is lower, but nevertheless clearly recognizable, the typical features here superimposed in the middle with the crystal structure. Now, what does this microcompartment look like? Now we zoom into one cluster here on the surface here. You see in red again the proteasomes. In yellow, you see CDC48. So, I mean, wherever these clusters is, ribosomes are excluded from the surface of the endoplasmic reticulum. And CDC48, I mean, tends to be located here more, I mean, you see it now here in yellow at the periphery of these clusters. But you see it's not a very abundant molecule here. So that is, I mean, the visualization of an ERAD microcompartment. We can do again a little bit deeper analysis. Of course, we can again look for the assembly states. We can infer from the conformation the different functional states. So ground state, substrate processing. Now, it's typically only something like 20% or so of all the proteasomes are actively degrading at any point in time. But if you look at the population that is next to the membrane, shown here in the histogram at the top right, there I think the fraction of proteasomes that are actively degrading, as judged from the conformation, is something like 50%. And if we take subtomogram averages of that subpopulation, we see distinct masses associated with the proteasome, with one specific subunit. I will come back to that a bit later. So that is basically the summary of this part here. So we have, a typical ARAT degradation cluster here on the surface of the ER. I think in the last part of my lecture, I will talk about a project we have been quite busy with in recent years, coming through an ERC synergy grant. It's a collaboration between my colleague Ulrich Hartl. Ulrich is a protein folding specialist. He wants to know how do aggregates affect protein quality control in the cell. Can we boost the cellular defenses against aggregation? My colleague Matthias Mann, bottom left here, is a proteomics expert. So he wants to know what is the composition of neurotoxic aggregates and how does the cellular proteome change in response to toxic aggregation. So we are interested, what is the in situ structure of the aggregates and how do they interact with the cellular environment. I will begin with, that was the first project in this series of experiments, which is still ongoing. It's related to Huntingtons disease, a terrible disease leading to muscle coordination defects. Now, it is caused by an expansion of the polyglutamine repeat, the polyQ repeat. If that is longer than 36 residues, it's located here, in this exon here. If that is longer than 36 residues, I think the longer it is, the earlier is the onset of this disease. So we had a model system here. Basically, we used both HeLa cells, but we used also neurons. Now, I should very briefly say that this is a fairly complex workflow because basically, let's say a neuron is a large landscape compared with the field of view of a tomogram. The first step is always vitrification by plunge freezing, of course. Then we use cryofluorescence microscopy to identify and locate, neurotoxic aggregates. We can transfer the coordinates and then do the focused ion beam milling in a very targeted way. And then we have to transfer things, of course, to the microscope. So what you see here is we have now focused on one Huntington aggregate in a primary neuron of the mouse. In blue, you see the Huntington fibrils. They are something like 3 nanometers in thickness, very thin. In red, you see basically the remnants of the endoplasmic reticulum. Basically, the fibrils interact in a very destructive way with the endoplasmic reticulum, and as a consequence of the disruption of the ER, the ribosomes in green are liberated and accumulate at the periphery of these aggregate materials. It's important to note that in this particular case, basically no macromolecular complexes are found in the aggregate material itself. Now, I think the reason for destruction of the ER are not fully understood, but whenever the fibrils, get into contact with, a membrane and even, a lipid vesicles, they change the curvature to an extent that the membrane becomes destabilized. The second case is, I think, in many ways more interesting. It's a system recapitulating, another disease, amyotrophic lateral sclerosis or ALS. Basically, the hallmark of that is a hexanucleotide expansion in this gene here. I don't want to go into the details. That might lead to a loss of native function of the gene product. But what it does also, it translates into the production of toxic D-peptide repeats. So inclusions in patients suffering from these diseases almost all contain polyglycine alanine repeats, as shown here. And I mean, they again then form aggregates. Now, again, the workflow is the same. We grow our neurons directly on EM grids. We identify the neurons that carry the aggregate material with green fluorescent protein, but it's also important to do the same in the absence of GFP, because GFP modifies the architecture of the aggregate material. Now we take the tomogram here of a typical polyglycine alanine aggregate, again in a neuron. At a glance, it looks we have fibrils, but they are not really fibrils, as you will see in the next slide. There are more ribbons, polymorphic ribbons, often bifurcated. But you will see on the raw data, if you look very closely, that we have numerous particles sitting in between the fibrils. Now we can do again the molecule by molecule analysis. So you see in red first the aggregate material. You see that as in Huntington, ribosomes shown in yellow never penetrate into the aggregate material. Chaperonin trick, which operates downstream of the ribosome and assists in folding, also is excluded from the aggregate material. There's only one molecular species that is massively accumulated in the aggregate material, and that is in green, the 26S proteasome. We have locally here something like a 40 to 50-fold increase of the concentration of proteasomes. Now, given that we have so many proteasomes, we have no limitations really in the number. We can obtain decent structures. I mean, on the 10 angstrom level, it's going down now to 7, 8 angstrom. We can very clearly distinguish in green those in the ground state, in blue those in the substrate processing state. What you see here in the substrate processing state, we always have additional mass not accounted for by the proteasome in magenta here. And when we map back those, I mean, identified as being substrate processing, they're almost in direct contact with the aggregate material. What happens here is basically that they try to degrade the aggregate material, but the glycine alanine repeat have a silk-like architecture, and that cannot be unfolded. And so as a consequence, the proteasomes become stalled and dysfunctional. Now, I think we are still improving that, but here you see that we can, in magenta again, the aggregate material, it interacts always with one very specific subunit of the 26S proteasome called RPN1. Now, comparing the two systems, this is from a News and Views written by Wade Harper. On the right-hand side, the Huntington, I mean, is destructive, and certainly that is not healthy. I mean, by damaging the endoplasmic reticulum. I think the mechanism seems to be completely different in the case of alanine glycine repeats. Basically, it's a kind of a trap or sink for proteasomes, and that compromises basically the fundamental protein quality control system of the cell here. So that is a different effect. I think one last, I think if I have 2 or 3 minutes, one measure, but I mean by incremental increases. I think I've summarized here a few issues that would help. I think we are moving from standard EM grids to micro-pattern grids for cells, for site-specific cell adhesion that will increase throughput. FIP is currently a terribly slow method. It will become faster, something like tenfold in the near future. We have to perfect the lift-out technology. Currently, the workflow is complicated because, I mean, the cryofluorescence microscopy and the focused ion beam are separate instruments, and the transfer is always a bit risky. So there will be integrated super resolution cryofluorescence, light microscopy, ion beam instruments. I think a major issue is beam-induced specimen motion. I think we have to work on that experimentally and computationally. I think cameras have improved massively. There's still a little bit of room for improvement. I think we need to develop better methods for mining the rich information of tomograms based on deep learning for segmentation, template-free methods for particle detecting and sorting. Last but not least, I think similar to what happened in the single particle field, we have to integrate complementary information such as proximity. We have to develop in vivo cross-linking mass spec for, generating restraints, guiding the modeling. So finally, this is the list of people. I think I don't want to go through all of them now, who worked on these projects in recent years. And I thank you for your attention. Can you hear me?
Yes, Professor, we can hear you. Thank you so much. So do you have time for some questions?
Yeah. I think we have to change now. Okay, I think I have already used the escape. And
Jetzt müsste irgendwo Präsentation beenden stehen.
Nee, das steht hier nicht.
Wenn man jetzt einfach die Präsentation zumacht auf dem roten.
Can you see me and hear me?
Yes.
Yes, we can see you, yeah.
Okay. I cannot see anybody, but it doesn't matter. Anyway.
So, Professor Baumeister, here's Kildare Miranda. I'll be handling the questions, okay?
Just one second. Okay, I think let's leave it like that. I think it's not important that I can see as long as you can see and hear me.
Okay, so shall we start?
Yeah.
Okay, so the first question came from Professor Vanderlei. Vanderlei, maybe you could ask him directly.
No, you can talk. You can read.
Okay. So the first question is if you did you find changes in the number, location, and functional state of the proteasome during the cell cycle in Chlamydomonas?
They're still trying to.
Okay, dann drücken wir hier eben nochmal.
Saiu?
Acho que ele saiu da reunião, é. Voltou.
Okay.
Now we can't hear you. You have to activate your microphone.
Oh, sorry for muting.
Okay.
Can you hear me?
Yep, now I can hear you.
Okay, so the first question comes from Professor Vanderlei de Souza. So he asks if did you find any changes in the number, location, and functional state of the proteasome during the cell cycle?
Yeah, there's constant flux of proteasomes into and out of the nucleus. Okay, so but we have not looked for that systematically yet. Proteasomes have nuclear localization signals, etc. And under certain stress conditions, there's a lot of import of proteasomes into the nucleus because the nucleus is strangely and surprisingly a major degradation compartment in the cell. So, but we don't see that really in Chlamydomonas, but we see that in yeast and in other eukaryotic cells.
Mm-hmm. Okay, thank you. And the second question is, did you find proteasomes in the mitochondrial chloroplast in Chlamydomonas?
No, they are just in the cytosol and in the nucleoplasm. Nothing inside. I mean, no.
Okay, and another question from Professor Marcia Chias. So does this location of proteasomes close to the nuclear and the R membrane mean that they are more active in screening defective proteins for degradation, just as they are synthesized then degrading old proteins?
Yeah, I mean, proteasomes degrade on one hand, I mean, misfolded proteins, they defective proteins, but they also degrade, of course, proteins which are no longer needed during some stages of the cell cycle. So, I mean, they also degrade, of course, healthy proteins which need to be removed in order to move forward in the cell cycle. Yeah, I'm not sure I answered your question now.
Removing the no longer needed proteins, and this screening action of just checking about misfolded proteins just as they are synthesized, that's something I just learned. I'm very happy. Yeah. And congratulations on your talk, and I'm very happy because it was both very beautiful and I have learned a lot of things.
Okay, thank you. Yeah. Now, I mean, for instance, in the ERAT microcompartments, it's very clear it's all misfolded proteins which need to be unfolded and are exported into the cytosol. I mean, there is a discussion, I mean, whether, CDC48 and the proteasome act on those being retrotranslocated sequentially. I think our data indicate that the proteasome itself is actively involved in extracting the misfolded proteins from the ER. So we think there is both a direct and an indirect ERAT mechanism here.
Okay, thank you.
Yeah.
So another question coming from Professor Marcos Farina. So how proteasomes move inside the cells? How do they move?
That is a good question. There are in neurons, I mean, they can be tethered to microtubules, and they use, microtubules as a railway system, but there must also be other mechanisms that is very specific for neurons. So there is kind of an active transport system in neurons, but that is not a mechanism. I think what governs basically the distribution of proteasomes inside the cell, let's say the formation, of these microclusters, is more dependent on phase separation mechanisms than on any active transport. But active transport exists for sure. Hermann Steller at Rockefeller and others have shown that very clearly. But in the cell, I think a major organizational force is phase separation. We have, it's very clear if you look at the dynamics of these ERAT clusters. They are highly dynamic. They fuse, they are generated in novo, and this is all very typical for phase separation, which is, of course, an increasingly important principle in cellular organization. Mm-hmm.
Should it be controlled locally?
Yeah, I think there is indication that polyubiquitin or tetraubiquitin can drive phase separation. So, the first step of ubiquitination and an accumulation of let's say polyubiquitin can drive, a phase separation that concentrates then also the proteasomes.
So talking about phase separation, so you mentioned this microcompartment, so my question is that we have this classical notion that compartment is our. Is there a feeling about that?
No, I think that is, I mean, there are other processes in the cell like autophagy, for instance, I mean, which involves, I mean, in order to initiate and I mean, involves phase separation. I think it's a new paradigm in cell biology, and I think it was met with a lot of suspicion maybe 5 years ago, but I mean, people are now convinced that phase separation is a crucial organizational phenomenon. The problem is always to identify the driver of phase separation. That is usually the big challenge. I mean, it's easy to say it's phase separation, and we see, I mean, let's say at the beginning of autophagy, I mean, before, I mean, the autophagosome is formed, I mean, basically a region which is not membrane bound, but excludes completely ribosomes, for instance. So, I mean, there's a completely separate phase, okay? But the challenge is really to identify who is the driver.
But should we call it compartment, since we don't have membrane?
Yeah, I mean, it's called a membraneless compartment. That is the common terminology.
Yeah.
Okay, so another question from Viviane Valença. What defines the proteasome stage? And
Pardon, what?
What defines the proteasome stage and the organization around the nuclear pore?
I think in that case, it's very clear that, I mean, the ones that are in the vicinity of the basket of the nuclear pore complex, there's a very distinct tether that tethers it to that. Again, I mean, we have candidate genes for this tether, but I mean, there is still, I mean, the knockout experiments have to be done to see, I mean, whether that changes something. I think at the periphery, it must be intrinsic membrane proteins, which, I mean, again, very similar to the situation on the endoplasmic reticulum, I mean, lead to the binding of the 26 process. I think in the case of the nuclear basket, in that subpopulation, we see structurally very clearly.
And Professor, from the technical point of view, you mentioned that data mining and the image processing is a great part of the work that is done now. So do you think that this is a profession, we have to have professionals integrated to to our facilities, dedicated to this part of work?
Yeah, I think that is still, in the case of tomography, somewhat underdeveloped. I think much of the segmentation is still done by many people manually, and I think that is very unsatisfactory. It's not only tedious, and it's also, I think, if you do it manually, there's always a bias. I mean, you want to do it in an automated way so that it becomes reproducible. I think the second aspect is, of course, I mean, basically classifications up tomogram averaging. I think we have to get away from template-based methods, and I think that will develop. I mean, the Chan Zuckerberg Foundation is extremely keen to invest massively in the development of more powerful algorithms for both segmentation. There's something commercially available from, I mean, like the Amira system, but again, it's not really tailored to the needs of Cryo ET, because basically that is is more for high contrast data coming from serial block phase imaging and that sort of thing. So I think a lot of progress can be expected from, improved methods for mining the rich information of tomograms. I think it's absolutely, I think you have to do both. We have to try to improve the quality of the data, which makes the analysis easier, but also, but that will be a relatively slow process, or to develop more powerful image analysis techniques. I think it's, if you build a facility, you absolutely need that.
Okay, great. So, are there any more questions?
Yes, good. I have one question related to localization of the proteasome inside the nucleus. Did you see association with the nucleolus, that is an active site of formation of ribosomes?
No, we don't see proteasomes in the vicinity of the nucleolus. I mean, we have just completed a study of nucleoli, but I mean, basically, this is the site, of course, for the biogenesis of ribosomes. And we can see very nicely how in kind of waves on the nucleolus, I mean, we see basically spatially resolved the biogenesis of ribosomes there. But inside the nucleolus, I mean, there's too much heterogeneity, but I mean, basically on the surface, we have very well-defined waves of ribosomes at different stages of maturation, but no problem.
Microtubules during division, because the microtubules disappears, and nobody knows exactly how it disappears. A simple disassembly.
We have not looked for that at all. I mean, as I said, I mean, the field of view of a typical tomogram is very limited at this moment. I mean, you must keep in mind that, let's say, even a tomogram 8K by 8K or so of data is a little bit less than 1% of the volume of the cell, okay? Yeah. So in order to cover the entire cell, I mean, that is so, I mean, there are ways to get that done. I think increasing it 100-fold is, I mean, maybe possible. So stitching tomograms and I think speeding up the data acquisition, which is currently, let's say, in the range of, let's say, it depends. Let's say 20 minutes, 10, 20 minutes per tomogram. I think it will go down to something like less than a minute. And then I think one can systematically stitch together tomograms and cover, I mean, the entire cellular volume. But currently, we look typically at 0.3-4% or so of the volume of the cell. I mean, so we have to zoom in on what we're interested in.
Yeah. Okay.
Okay. Any more questions? So if there's not any questions, so thank you so much. Thank you again, Professor, on behalf of our cell biology and parasitology program and our institute. We thank you very much for your talk and for your time.
Okay.
Vanderlei, if you want to say a few words.
No, thank you very much, both to accept our invitation, and I hope to see you personally very soon.
Okay, let's hope for the best.
Okay. Thanks again, Professor. Bye-bye.
Bye.